have done it was the Exodus. Am I on, Mike? Good evening, everybody. Very warm welcome to Wesley Methodist Church for the latest in our series of talks on the general theme Science Meets Faith. My name's Malcolm Hazlett. I'm one of the stewards here at Wesley. Apologies from our superintendent minister, Kevin, who's been called to a high-level clerical meeting in Norwich, and Professor Judith Liu, who would normally stand in for him, is at an even higher level conference in Lausanne in Switzerland. So you're stuck with me, I'm afraid. And I'm a mere linguist, I'm not anything scientific. But we look forward to hearing from Dr. Rimmer. I should underline, first of all, be careful of the tripwire, which is probably a health hazard. A few householding things. Toilets are over there for those who don't know the church. Gents that side and the ladies there. If by any strange chance there is an alarm bell goes off, you exit by that or that or that, and all those doors are in fact open and should be able. And I think we are supposed to gather in the bit of Christ's peace next to the church, just over there. But that shouldn't happen. It's never happened in my experience, and I've been here 10 years. Right, now I had somewhere a bit of paper, yes, because I just wanted to quote. I think everybody knows what the Cavendish Laboratory is. I just thought I'd quote from there website an interesting little quote which says, since 1874, the Cavendish has been at the forefront of scientific discovery. The electron, the neutron, the structure of DNA, 31 Nobel Prize winners. And these past successes spur us on to continually redefine and expand the frontiers of physics and its role in the world, passing on our expertise and instinctive curiosity from one generation to the next. And also I'd like to just quote from something Paul gave me himself, about himself, just to introduce him. He can introduce himself a bit later. Paul Brandon Rimmer is an assistant professor of experimental astrophysics at the Cavendish Laboratory, University of Cambridge. He's building a star in a lab, and he uses that lab to search for comfortable places in this universe to start new life. Paul's married and the father of 2 sons. He's a planetary astrochemist by training, a Christian by religion, and a Platonist by philosophy. Perhaps he could explain that last point. Also, could I just say that to cover expenses, we do ask if you can afford it to give a small donation. There's a box there. It does say miscellaneous stationery on the box, but it's not for that. It's for any donations that you care to give. And the money we receive, we cover what expenses we have, but we also like to give the rest to a charity. And Paul has chosen as his charity Spectrum. Now, he will have to explain exactly what Spectrum is, because we're not quite sure.
Okay.
So anyway, without further ado, I will pass it over to you, Paul.
All right. Thank you very much. I might need to be prompted again about Spectrum. I can explain what it is at the end. I think a brief statement will suffice. But mostly I'll be talking about this. Are we clueless about the origins of life? So I'm going to start with a question. How do you know a puzzle is solvable? So I bought this puzzle this morning. That's probably a good sign. This is a jigsaw puzzle. It comes wrapped. That's also a good sign. Probably all the pieces are there. I could, of course, have found this puzzle maybe at a charity shop, or even worse, in one of my storage rooms. There could have been a hole in the side. Maybe I found a few pieces scattered around. Then I might be more concerned whether the puzzle is in fact solvable. I could try to solve the puzzle by what I will call a brute force method. I know this is a 1,000-piece puzzle, and maybe I know the rough shape of it. So I could just try a random piece in one corner and then go all the way through and find out that doesn't work. I could try another random piece, again, all the way through, find out if that works. And that would be about 10 to the 2,000 combinations. So that's one with 2,000 zeros afterward. There are people who do seem to solve jigsaw puzzles that way and don't make a lot of progress. And I do have to admit, I'm not the best jigsaw puzzle solver, but I can do better than that, because that would require much, much longer than the age of the universe. So, origin of life is a little bit like this puzzle, except that we have different pieces. So I'm going to pull out some of these pieces. They might come out at random from my bag. So this is a nucleotide, a nucleobase, a sugar, and a phosphate group. And this could be arranged a whole bunch of different ways. There are a bunch of different bases we could have. Even with just these same atoms, there are a whole bunch of different ways that this could be arranged. How many different ways? Well, it depends on how bonds are made, and I'm just going to explain that just a little bit. So it's all about electrons. Hydrogen makes 1 bond. Oxygen makes 2, generally. Carbon tends to make 4 bonds. Nitrogen tends to make 3. Sulfur is promiscuous a little bit in this regard. It's got lots of electrons, and sometimes shares deeper in deeper shells, and so it can make 2 bonds, or 4, or even 6. Phosphorus, similarly, it can make 3, it can make up to 5. In some settings, it can make even more bonds. And so I can think about how I might rearrange all of these. And if I constrain myself to relatively stable structures, which really helps a lot, because there are a lot of different ways I could put this together, a crazy number. If I relegate myself to stable structures, there's about 20-ish isomers. So 20-ish different ways I can arrange this where it would still be pretty stable in water. Same with, let's see what else I can find in here. This is alanine. This is one amino acid. It has an amine. It has a hydroxylic acid here, so these 2 oxygens and a carbon. And then these carbons, this is a side chain, so it would be different for every amino acid. All amino acids look the same in this respect, but have a different side chain. I could change this already by just, if I wanted to, pulling this off and putting this here and putting the hydrogen on the other side. That would be its mirror image. Most amino acids have a distinct mirror image, which is a completely different molecule. Try to fit that back on. And again, believe it or not, about 20 different stable forms for that. I'm not going to try to talk too much about this part of the puzzle. This is a really important part, though. This is a phospholipid. So you have this thing here, which has negative charge and positive charge. It's polar. It attracts water. This part is apolar. It repels water. And so these, if I put this, not obviously the model, I would need like 10 to the 23 of these, so one with 23 zeros afterward, but I would need a fair number of these put in water. They will naturally make a sphere, because all of these parts will find each other and stack together, and all of these parts will find the water on the inside and the outside and stack together, and they'll make a sphere just naturally. I can put them in. This, by the way, I ran out of carbons, unfortunately. This is actually quite a bit longer, and there's a double bond down here somewhere. There are various forms of these. Life uses thousands of different ones of these. So I'm just going to set those aside for now, actually. That's talk about complicated. And because it uses so many different ones, there might be many different combinations that would still work. It uses about 4 of these. There's deoxyribonucleotides and just the ribonucleotides. This is a ribonucleotide. It's adenosine monophosphate. There are 4 different letters, maybe that you've heard in DNA and RNA. This is one of the letters, A. And then there's about 20 of these. I'm just going to concentrate on these. So 20 different versions of this, 4 different versions of this, 20 different ways to arrange each of them, and about, a few million of them in a very simplified version of E. coli, a very simple sort of cell, simple. If I arrange all of them in all of these different ways, that gives me about 10 to the 2 billion combinations, which is a lot more than the 10 to the 2,000 combinations here. And the thing is, if I really want to use the puzzle analogy, I need to go even further. I need to say, I actually have to put the puzzle in the right order. So I would have to choose a particular piece here to start with. It might be a middle piece. Then I have to choose an edge piece, everything else like that. That will actually increase the number of combinations from 10 to the 2,000 to 10 to the 4,000. That's one with 4,000 zeros. You say, well, that's just double. That is not double. That is, if winning the lottery was a 1 in a million chance, or say, a 10 to the 6 chance, 10 to the 12th would be winning the lottery a million times in a row. That's hard. It's just a ton of combinations. And the reason why I say it has to be put in sequence is, imagine that I just say, all I need are the ingredients, and then I have life. Well, then I can take a living cell, I perforate the membrane, I let it all leak out, and I say, great, that's a better prebiotic soup than almost any prebiotic soup that would have existed on early Earth. It has all the enzymes, everything else, and then I want to wait until that becomes alive again. I will be waiting a very long time. It's not going to become alive again. It's dead. The sequence reflects how it's not just the chemistry, but the behavior you need to get. There's a particular kind of behavior. So the sequence really matters. The sequence here matters as well. So now I'm going to leave this as an open question, which I'll come back to at the end and encourage some discussion about. How do I know when a puzzle is solvable? How do you know when a puzzle is solvable? How much of it depends on your ability as a puzzle solver? How much of it depends on the person who made the puzzle? We'll come back to that. Just a quick thing about representation. I'm going to be drawing some chemistry here. Please feel free to ask questions about it. I don't even mind questions during the talk, actually, but certainly afterward. But just to show, this is kind of like this. This is actually a precursor to this. You can see all the different bonds. They're all written out where the white ones are the hydrogens, and the black ones are the carbons, and the blue ones are the nitrogens, so the N's, and the red ones are the oxygens, so the O's. And that looks like a mess. So we can simplify by just collapsing all the carbons onto vertices and just suppressing all the hydrogens. We know that carbon needs to make 4 bonds, so whenever we get a particular place, I can't quite highlight it, but you see that OH on the top, and then you go down and there's the C with the 2 H's. Well, I see it has 2 bonds, so I just add 2 extra bonds for those hydrogens. So when you see structures like that, you have to kind of fill in the blanks. It simplifies life. So one of the things I'm going to say, which you've already probably figured out from the combinations we need to look through. I'm not saying something as simple as this is how life started, although there are people who think that this is how life had to start. None less illustrious than Jacques Monod, who was one of the founders of molecular biology. He felt it was a complete accident. Things just came together, there was life. It will never be reproducible in the lab. I mean, look at how many combinations. And so it's a waste of time to research it. There are people of very different perspectives, including faith perspectives, who share that opinion. That's an extreme opinion. But I do think we need enough epistemic humility. This is humility about what we know and what we don't, to just say, I don't know what the right answer is. When I meet another researcher and they have a different idea, my primate kind of attitude is to fight, and so I want to say, you're wrong because that's not my idea. It's best to let them explore their own way. Let me explore mine. And unless there's a good empirical reason to rule it out, I shouldn't try to convince them to stop exploring, and hopefully they won't try to convince me to stop exploring in my direction. And that includes people who are skeptical about this. This is a couple papers listed here that talk about doing things in that direction. I think it's a really healthy direction for science, and also for communicating science and just talking about this issue generally. So I'm going to show a few quotes of different people who have different perspectives on this. Here's one. Origin of life as a field as a whole is unconvincing, generating results in toy domains that cannot be scaled to any real-world scenario. I've talked with this guy, and he says, actually, no progress has been made in origins since Miller, which happened in 1950s. We'll talk a little bit about the sort of history. And this is William Baines. He's a leading astrobiologist. He does beautiful work at MIT. He's an atheist. So I chose these quotes specifically to anti-correlate with the way you might think certain religious attitudes are. He's a complete skeptic about origins. He doesn't believe in God. We're moving to less skepticism. This is viewed in this way, so in the way of what we know about prebiotic chemistry. The prebiotic synthesis of building blocks, to which we have devoted so much of our time, only corresponds to a small increase in complexity of the system and no increase in its aliveness, which is a very humbling thought. So we haven't made a lot of progress toward our real goal. That's John Sutherland. He's a leading organic chemist who works in this field. A lot of his chemistry is the chemistry I'll be talking about later. We have a more optimistic view. You start with hydrogen cyanide, you mix that together with small sulfur-bearing molecules in water, and you end up forming all the building blocks of life that exists today. That's Karen Oberg. She's a leading researcher also in this field at Harvard. She's a Catholic. And finally, this is the most optimistic that I can find anyway. You start with a random clump of atoms, you shine light on it for long enough, it shouldn't be so surprising you get a plant. And that's Jeremy England, a physicist, by the way, not a chemist, so that might be part of it. He is also an Orthodox rabbi, as well as having been a professor at MIT. So which one of these is right? I don't know. I tend to lean a little bit more toward the sort of John Sutherland picture. There's just a lot we don't know yet, and there's a lot to learn. I think there are a lot of interesting things we've been able to learn along the way, but to do that, I need to make a few assumptions. One of them is that life is made of chemistry. I'll quote C.S. Lewis right now, or paraphrase him, when Eustace was talking with the star in the Voyage of the Dawn Treader. He says, "I thought stars were made of gas and dust," and the star says, "Even in your world," I thought stars were gas and dust. And the star says, "Even in your world, that's not what stars are, that's what they're made of." And it's the same sort of thing here. Life is made of chemistry. That doesn't mean life is chemistry, and that's a really important distinction. But life, I am going to assume, is made of chemistry. If you disagree, a lot of the rest of this talk isn't going to make a lot of sense. The origins of life, at least life as we know it, is at heart an organic chemistry problem. That means these carbon-hydrogen bonds and the other bonds that the carbon makes is really important. That's the core. There's a lot of other components. I myself am a physicist who works in astrophysics, so that's an important component. There's a planetary science component, a geoscience component. Clearly there's a biology component too, although we haven't gotten quite far enough for that to matter too much yet. But at the core, at least at present, it's an organic chemistry problem. And I'm also going to assume life started on Earth. I don't know that for sure, and in fact, a lot of my arguments don't depend on that. If you want to think life started on Mars and then hitched a ride or something, most of what I say will still make sense, so that's just a weak assumption. So I brought out a handful of molecules here. I'm going to go back to them a little bit. Life as we know it needs four things. It needs a container, and we've already talked about that's what this does. It makes the container. And it needs a container for a variety of reasons, I think. It needs it to individuate, so there's something to evolve. There's some way to contain the information and differentiate it from information of other individuals. I think it's important because it concentrates chemistry, and it's important because you can build gradients, differences along those boundaries that can be used for energy, and that's the way all life as we know it gets energy. It needs some informational molecule. That's what these guys do. So ribonucleotides, deoxyribonucleotides, the information molecule needs to link together. It needs to store and transfer information so it can be copied, so you can have replication. And the storage and transfer of information needs to be exceedingly accurate, but not perfectly accurate. It needs to be able to make a few mistakes so you can have mutations, you can have Darwinian evolution. But if it's not very accurate, then my kids won't look enough like me. There will be no inheritance, and so nothing can be selected. I need some way to control the chemistry. This is really important. If just random chemistry ends up happening, I just get a mess. I don't get life. This is largely done by these amino acids when they link together in chains of hundreds or more sometimes, but as short as chains of 5 can be useful, into proteins, largely. RNA does a little bit of that too. And then I need some sort of system by which I can convert energy to these large molecules, or I can get other large molecules like sugars, which I did a little earlier today for dinner, and make that into energy, which I can then store and use in different ways. And there needs to be a sort of progression. I can know a little bit about where I might start, small molecules that are likely to exist on a planet, progressing to larger and larger molecules that link together, and importantly, have the behavior that's a bit lifelike. Eventually, I might get to a protocell, something that can make copies of itself, contain itself, control its own chemistry, but will be much, much simpler than E. coli. So way, way far from what's often called the last universal common ancestor, or LUCA. This is important. I'm now run into two other epistemic challenges, by the way. One is, LUCA, the last universal common ancestor, is as far as these evolutionary methods can really get. They can get a little bit before LUCA, actually. There's a guy at MIT that can trace different genes and figure out their expressions and trace to a few million years before or a few million generations before. It's really hard to get far before LUCA. the last universal common ancestor. And that's pretty much as complicated as a bacterium, or roughly as complicated as one of my cells. My cells are a little bit more complicated in certain important ways, but compared to this protocell, very complicated. So we can't learn a lot from Luca. There's already been millions of years and billions of generations of evolution that separate Luca from this protocell. Also, there's no rocks that go back this far. This is around 4 billion years ago. We don't have a record, on Earth anyway, for what this was like. We have a little bit from minerals and a little bit from other hints. Interstellar objects, Mars, certain other places give us a bit of hints. But it's really not well constrained. But we do know about what chemistry life uses, and we can use some of the chemistry that we think would be present on early Earth to see what could happen in the lab. And it's surprising how much we can learn from that, actually. So, because it's a relatively small crowd, I am going to risk a little bit of audience participation and just ask anyone who wants to show their hand, how would you link the molecules of life together if you were going to do it? How reactive would you make them, and how would you make them react, do you think?
I'd probably think of something that's pretty similar in temperature and pH to what we have.
Yeah. No, I would do the same. Would you want them to be very reactive? Would you want them to be somewhat reactive or not very reactive?
Probably in the middle.
Probably somewhere in the middle. Yeah, no, so middle of the road is good. Yeah. So that's actually a really good point. Was there another, oh, please. Okay. That was actually almost word for word what I was going to say. Yes. Oh, yes. So the answer was we want them to, and I'm going to paraphrase a little bit because that's the way that my memory works, is we want them to react when we want them to react and not when we don't want them to react. We want them to react selectively, and that's exactly right. And life does this in an amazing way. Almost all of its reactions are thermodynamically prohibited. They happen in water, so they're dehydration reactions. They're reactions that give off water in water, which nobody, no molecule wants to happen. I'll illustrate this with, going back to our amino acid, our amine group right here, our carboxylic acid over here, and our side chain, which is the one that can be different. The carboxylic acid will react with that ammonia to form what's called a peptide bond, and it releases water. Everything in the environment wants it to move the other way. When you already have proteins, those proteins, what they do is they hold these molecules together, not so much with the amino acids. It turns out largely ribonucleotides do it here, but it's the same idea. They hold the molecules together to react, and then they surround the reaction site, remove the water from the reaction site to make it favorable to react. Once it reacts, it goes off. Eventually, it does degrade. Same thing, by the way, with ribonucleotides. I'm just going to point out again. Nope, that's the really big one. A base. I want to hook this different base to the same sugar, and then to the phosphate, and then the phosphate hooks all of these things together. So you can imagine a bunch of these things in sequence, like what's shown there. That phosphate and oxygen link everything together. Every single step along the way, dehydration. Hooking the sugar to the base gets rid of water. Hooking now what's called the nucleoside to the phosphate gets rid of water. Hooking these two together gets rid of water. All of them are thermodynamically unfavorable, and when we don't have enzymes, what this means is water is a really hard solvent to work with. Really important, by the way. In fact, if people were thinking about life in other solvents, I would encourage them to look at molecules that don't react much, because you want the reaction to be selective, and if they react all the time, you just end up with a mess, not with life. I'm going to break a little bit into just a brief history of the field to talk a little bit about what progress has been made both to understand these very things I've been talking about and also where the field's been going in trying to understand how to resolve these particular problems. And it turns out this pretty much does start with Darwin, at least as far as I can tell. I'm very happy to find, if anybody knows anything that happened before Darwin, I'd love to know. But Darwin's 1871 letter to J.D. Hooker, as far as I can tell, kind of started all of this off. So he says, "It is often said the conditions for the first production of all living organisms are now present, which could ever have been present. But if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric acid," I'll start pulling some more molecules out there, molecules like that, phosphoric salts, rather, light, heat, electricity, etc. present, that a protein compound, one of those things like amino acids, was chemically formed, ready to undergo still more complex changes. At present day, such matter would be instantly devoured and absorbed, which would not have been the case when these creatures first formed. So this is the first kind of idea, but this is just an idea. It's a brilliant idea, and in fact has a bunch of important insights we could talk about later. It's amazing how many insights are built into that little statement. But it's just an idea. And then there were people who came along and built on that idea with just more ideas. Just ideas. No experiments, no observations. One of them is Alexander Oparin, and the other is John Haldane. Around 1924, 1929, they figured out about how when our Earth formed, it formed in this nebular gas. The nebular gas is mostly hydrogen, so they pictured a gas that would mostly have these kinds of species: methane, ammonia, molecules of that nature. I'll get rid of these bigger molecules here. And both of them had the same idea pretty much independently. Oparin developed his a little bit more. Stanley Miller and Harold Urey worked together. Miller led this particular experiment. And his single-author paper in 1953 ended up finding out that if you did start with that kind of atmosphere, ammonia and methane with water and hydrogen, you end up forming amino acids. You end up forming glycine, you end up forming alanine, the one I just showed you. Small concentrations. He doesn't know how they formed, that's another problem. And he doesn't know whether the gas he used was the correct one. Yet another problem, but he forms a few of these. And it's kind of coincidental that this happened at the same time we found out, or started to find out, how complex life really was, because at the same year, Watson and Crick, building on Rosalind Franklin's beautiful observations, were able to infer the structure of DNA. There's a nice paper by Aaron Klug that talks about Rosalind Franklin's contribution to this. Turns out life is very complicated, actually, and not just a handful of amino acids. You need nucleotides for one thing, and nucleotides are not formed by this chemistry. This chemistry, however, does form a lot of other molecules. I'll pull a couple of those out. Cyanoacetylene, this weird-looking molecule, is one of the main molecules that's formed, and hydrogen cyanide. Seems horribly toxic, but actually those are really important for all of the subsequent chemistry. Leslie Orgel built on a lot of this sort of chemistry and tried to combine the worlds of Crick and Miller. And he basically looked a lot on single reactions. One reaction happening with another makes a product. He then concentrates that product, does another reaction with that product, goes on, builds up complexities, tries to understand the basic chemistry. At the same time, he starts working with what are called ribozymes, which is RNA that does the kind of work of proteins. So maybe just RNA starts, so Orgel is a big fan of the RNA world model. Around the same time, a patent lawyer, it's always patent clerks and patent lawyers, isn't it? It's Gunther Vectorsäuser, got involved. I don't know the entire story about how he got involved, but he got involved in molecular biology, got very interested in the Krebs cycle, the citric acid cycle, and saw how hydrogen and sulfur and iron play a particular important role in that cycle. And his ideas eventually become what's the hydrothermal vents hypothesis, basically, and certain other hypotheses is called metabolism first. So he starts with the metabolism, Orgel starts with the informational molecule. I don't have enough time to talk about all the other people who, every one of those molecules somebody starts with and says, that's the one. Eschenmoser says, well, first of all, maybe it's all of them. And Eschenmoser also is one of the first people to really talk about epistemic humility. He says, we don't have the rock record, so we can't know. Luca, too advanced, so we can't know. We can't figure out how life really started, but we can figure out how life could have started. I actually beg to differ a little bit on that, depending on how common life is. We can talk about that. But certainly, with this approach, that's the best we can do. And he drew a lot of analogies between the molecules life uses and the kinds of molecules you see present on Titan, and again, the molecules that Miller and Urey were producing, especially these guys. And he also approached a sort of systems chemistry. Instead of doing a single reaction at a time, he started mixing things together, making it a bit messier, which actually made the chemistry much richer than people imagined. And he kind of started a revolution. I'm just going to show a bunch of pictures. I'll name the people. There's a story for everyone, so if you want to ask questions at the end, you're certainly welcome. I don't have enough time to go into all their stories. But there's been a huge explosion of new ideas and new directions in this field, starting from the top and then just working over. That's Claudia Bonfio, Dimitar Sasolov, Furkan Ozturk, Zoe Todd. Then there's John Sutherland. Then under John Sutherland is Sukrit Ranjan, Donna Blackmon, Karin Oberg, Jack Shostak, and at the very bottom there is Beitul Kachar. It's become a lot more diverse field in every respect, and it's also become a much more interesting and full field. And there are many more that I could have included pictures of, by the way. That's just highlighting a few. And so here's some of the things I think we've learned. So I'm now putting my cards on the table. I do think we are not clueless. I think these are the clues. There are some necessary conditions for this chemistry. It's disequilibrium chemistry. What does that mean? It's special. That's the way I'm going to say it. It's special chemistry that requires energy. What kind of energy? Maybe it's environment specific. Maybe it's general. It has to be high yield selective chemistry. That means the reactions have to produce the product you want and not the products you don't, as much as possible. I've already talked a bit about that, but I'll defend that a little bit more. And it has to be prebiotically plausible. What does prebiotically plausible mean? It means something that could happen on the surface of a planet without a chemist being present there making it step by step go along. So why high yield and selective? The arithmetic demon. Let's say I start with 1 mole per liter. That's 10 to the 23rd of the molecule I want in a liter. That's like a soup. It's pretty thick. Like, there's a lot of the molecule. Way more than you would expect in a lot of prebiotic chemistry. And I am going to make life in 30 steps. That's very ambitious. And each of those steps, I lose a little bit. That's what the yield is it says how much I retain. So 99% is almost all of it. I only lose 1% every reaction. 10% is I lose 90% of it every reaction. At the very top is where most prebiotic experiments live. So very high concentrations, because you can see things more easily. The synthetic limits, the limits of synthetic chemistry, I found a few synthetic papers that use micromolar concentrations. That's 10 to the minus 6 molars. So that's getting a little low, but possible. You run into a science problem when you go to much lower, because you get to detection limits. We just don't have an ability to see the chemistry. Maybe amazing chemistry is happening, but if we can't see it, we can't study it. So we need a non-standard approach if we start going below that. Below that even gets to 1 reaction a year, and then even below that is homeopathy. So that's, we have left science and we're doing something else. So there are a few ways around this. A few steps. Maybe instead of making life in 30 steps, we make it in 3. And if anybody ever does that, I'll be happy, as long as those steps are plausible, I'll be happy to concede they've discovered the origin of life, and that's a great solution. But for now, I'm skeptical of that for a variety of reasons. I mean, look at how complicated life is. So I'm going to cross off the few steps for now. A high yield for every reaction, I think, is also implausible, but not as implausible. We don't know enough about the chemistry. It's a huge combinatoric space. Maybe we'll find perfect chemistry for all the reactions. There's a third thing, which is workup. That's what organic chemists call when you make a product and it's mixed with a bunch of other products, and there are things you can do like distillation and other things to concentrate what you want and get rid of what you don't. Nature can do the same thing. If you have clays, it can select a particular molecule instead of another. There are certain ways in which the molecules themselves can act as catalysts. It's called autocatalysis. They can enhance their own production. There are certain interesting behaviors that chemistry can have that act like workup. I'm going to say that we need kind of both. We need fairly selective chemistry, as high yield as we can get, and we also need this workup. And this is really what, this kind of philosophy is what led John Sutherland's group to come up with this kind of chemistry, which is selective, high-yield chemistry that does some pretty amazing things. So right now, we have hydrogen sulfide, H2O, so it's hydrogen sulfide in water, along with the hydrogen cyanide. So the hydrogen cyanide is one of the key molecules. The hydrogen sulfide in water will naturally disproportionate. It depends on the pH, but at good pHs for this chemistry, some of it will break apart like that. And ultraviolet light can come in, knock that electron off. And that electron can go back onto the HS dot now, make HS minus again, nothing happens. It can also go onto the proton and make hydrogen, so you can get hydrogen. It can also attach itself onto the cyanide, and when it does that, the cyanide can attract a proton. This is now a nitrogen with 2 bonds. We said it had to have 3, and it can actually just pick up a bond right off of that H2S. And what happens here is this is an imine, and that imine will hydrolyze. It means it just reacts with water very, very quickly to release ammonia, another molecule we're interested in, and it makes formaldehyde, almost a sugar. We're almost at a sugar. And the same sort of chemistry, this is the very first reaction I ever performed in a lab. I'm not a chemist by training, but I got to work in organic chemistry lab. This is the very first reaction I performed, is formaldehyde and cyanide. Cyanide can also break apart like this, disproportionate, and it can react to form glycolonitrile. Very fast reaction. Same triple bond carbon, same thing can happen, triple bond carbon nitrogen, same thing can happen again. And that gives you glycolaldehyde, which is a simple sugar. And amazingly, depending on what else you have around, you need phosphate for one. Phosphate helps to control the pH, and it removes a lot of the side reactions. You need ultraviolet light. The UV light helps to clean a lot of this chemistry. But depending on what else you have around, if you have a lot of ammonia or hydrogen sulfide, you make amino acids. If you have that cyanoacetylene, you make the nucleotides. And if you don't have those, just the phosphate, you actually start making the lipids. So you get everything all in one pot. It's quite astounding. And it works very, very cleanly, and it's easy enough that even I can do most of the chemistry. There's a few of the steps I can't do so well. We can talk about those later. Maybe that's a sign it's not so prebiotically plausible. This is not the only possible chemistry. I'm not going to talk about the rest, but there are also very fascinating ways. All of them require cyanide and cyanoacetylene, by the way. Even this last one, which tries to do it without cyanide, but ends up making cyanide along the way. I think that's a clue, too. It's chemistry plus environment, though. Where does this happen? Where do we do this kind of chemistry? There is non-cyanide chemistry. Some of it is in underwater hydrothermal vents. That's a possibility. Surface hydrothermal vents I like, because I've been working on that. If you ask questions later, I can talk about it. Alkaline lakes are really interesting because they can concentrate that phosphate. Even today, some of these alkaline lakes have enormous amounts of this phosphate. Underground, radiolysis makes the hydrogen and can do some of this chemistry. Basaltic glass. It turns out that when you put some of these molecules on basaltic glass, and if they have a high enough energy, they polymerize naturally to sometimes 100-length polymers. Really fascinating stuff. Cosmic dust, it's raining down. It's mostly made of this carbon-nitrogen triple bond stuff, so it's kind of a polymer almost of cyanides. And giant impacts. Carl Sagan thought about these importantly. These can deliver things because comets have a lot of cyanide and cyanoacetylene and amino acids and lots of other things we could use. If we can get them gently enough to the surface, we could use them. There's another way giant impacts are important, and I'll talk about those near the end. But the main thing I'm going to just emphasize is this is another mess. Chemical parameter space is effectively infinite. We already talked about that. At least as far as I'm concerned, it's effectively infinite. The possible environments are exceedingly diverse, tons of different environments. But if you can't make and test predictions, it's not science. And I want to do science. The combination of environment and chemistry can be tested. So what I can do is I can find a good chemistry. It has to work in the lab first. If it doesn't work in the lab, give up. Find something that works in the lab. Say, what kinds of environments? Use your imagination. What kind of environment could that happen in? Find the sort of environment that can happen in. And then ask your friendly neighborhood geochemist what goes into that environment. Put that stuff in, see if it still works. A lot of this is about the initial conditions, a lot of what people do. I do something a little bit different. I've been inspired by Masanobu Fukuoka, a farmer who proposed this one-straw revolution, this do-nothing farming. The sort of idea of if you have a garden, weeds grow in the garden. He got frustrated with weeds. Everybody gets frustrated with weeds. He said, how can I make the plants I want to grow like the weeds? Find the behavior of the environment that overlaps with the purpose of the farmer. It's actually kind of controversial agriculture. I'm not going to try to defend Masanobu Fukuoka for gardening or farming, but I am going to say that this strategy is necessary for prebiotic chemistry. We need to find out where the behavior of the environment matches the purpose of the prebiotic chemist. Where can this chemistry happen on its own? And this involves a great feedback between the geoscience and the systems chemistry, not so much in just the initial ingredients, but figuring out how long it takes each reaction to happen, how long it would take reactions to happen in a more realistic conditions, and testing those by reproducing those realistic conditions in the lab, which is a lot of what we do. And for this, timing is absolutely everything. Can this prebiotic chemistry happen without a prebiotic chemist? Do-nothing chemistry is all about timing. And the scenario environment together determine the time scale, but if I had to test every single one, it would take forever. Even an army of chemists would just take way too long to do all of this. What we want are rate constants. These are numbers that tell us what would happen if the environment were a certain way. They're hard to measure, but once we have them, we can put them into a model. We can then run the model a billion times, figure out the best places where this chemistry happens, test those in the lab to make sure the model's still working, and then start making inferences for things that we can look for, say on Mars and various other places. I have a scenario. I'm running out of time, so I'm going to skip the scenario to just get back to the puzzle, actually. But if you want to ask questions about it, this is one of the things our lab has learned, which gives us the clean chemistry, the cyanide, the cyanoacetylene in large concentrations with none of the side products, and allows for pretty good timescales to do at least the first few steps of this chemistry. I also need to emphasize I'm not the only person who does the work. Science is about people more than it is about ideas. And this is my group. These are the people really doing this sort of work. Teresa Constantinow, Skyla White, Teresa Drobnik, Sai Morali, and Lucas Rosmanath. I didn't picture Richard and Katrina because they weren't there that day. I also collaborate with a lot of interesting people both in Cambridge and outside. So, hints are abundant. That's my argument anyway. And they're emerging at an accelerating pace. But we've just scratched the surface. This isn't life. This is just the building blocks and a couple ideas about how to link them together. What if this ultimately leads nowhere? I'll go back to the puzzle question. So there's a great history about this. There's a history and philosophy of science about this. Stanley Jackie has written a great book called Science and Creation about this very thing. There's also Dominion by Tom Holland, which has talked about this. I do think a Christian worldview is really healthy for approaching this. I'm not enough of a historian to argue all the history for it, but I will say that at least from my own perspective, if I had this particular view, which many pre-Christian, pre-Jewish, pre-Islamic views had, irrational animals in a meaningless universe, I don't know how to solve the puzzle, and the puzzle maker didn't know how to make one. I would be very concerned about whether I'd be able to solve this puzzle. We then transitioned into this particular view, especially during the Renaissance and the scientific revolution, rational animals in a meaningful universe. The puzzle maker knows what they're doing, and I have some hope, even though I'm a finite intelligence, for making some progress towards this. We then, during the Enlightenment, kind of went to rational animals in a meaningless universe. I would be concerned, no matter how smart I am at solving a puzzle, if I'm given a puzzle that was designed by someone who was wicked and wanted me to struggle with puzzle solving, or just someone who was incompetent and was just cutting random pieces, I'd be worried about whether I could solve it. My hope that I can solve this actually does come from my Christian faith, and I do think that a theistic faith is important for that sort of hope. Solving the puzzle won't solve everything. Scientific discovery is not like filling a chasm. It's not like God of the gaps. It's like an island. The more you learn, the more questions you have. The more the unknown becomes. And there are truths science just cannot touch. I'll finish with this one idea. That's a picture, by the way, of my son. When my son was was born, a new intelligence came into the world, a new first-person perspective. There are people who argue about irreducible complexity. I don't know about irreducible complexity. I don't find those arguments very compelling. But the first-person perspective is irreducible. You cannot break down the first-person perspective just into science. I have some very strong arguments about why I think that has to be the case. And the same thing is, even if you had a great predictive model for, say, which we do actually in large part, we know how conception happens, we know how child development happens. Just knowing that doesn't answer all the questions we're interested in. What is this person going to be like? What great things are they going to do? What decisions are they going to make? Same thing with crossing the Rubicon. If some very smart physicist said, oh, I can explain why Caesar crossed the Rubicon and why the Senate reacted the way it did and what happened with Rome, I have a model that captures where all the particles were, and I know the laws of physics, so I'll figure out where all the particles... I don't even need to know Latin. I can just figure out exactly what's going to happen. They'll have an answer that's predictive, but not very satisfying for really understanding the reasons why. And I think a lot of origins of life, aspects of it, are going to touch on that as well. Just real quick, here are some books if you're interested in more. Is Earth Exceptional? Prebiotic Chemistry. If you already have a sort of A-level knowledge of chemistry, that's a great book by Fehrenbach and Cleves. Is Earth Exceptional by Shostak and Livio is excellent for just public. If you know a lot of physics and are interested especially in the information problem, Manfred Eigen's written this brilliant but very thick book, From Strange Simplicity to Complex Familiarity. Some other references. I'm just going to emphasize Stacy Trasancos' Particles of Faith, which covers a little bit of both Tom Holland's book Dominion and Stanley Jackie's Science and Creation. It's a beautiful book, science popularizing, but from a Catholic Christian perspective. And then finally, I had 3 goals in this. My my least important goal was to convince you that there's a lot of hints for origins of life, so we are making progress. My more important goal is to just get you interested. Maybe you disagree, maybe you think no progress is made. Hopefully you're interested in the science, you can learn more. And no matter what you think, there's ways you can contribute. If you're a Cambridge student or Cambridge faculty or someone else with Cambridge affiliation, there's the Leverhulme Center for Life in the Universe. Come up to me after, talk with me. There's a lot of cool things going on in this very field. My main motivation is to reflect how my Christian faith gives me hope in a solution, and to hopefully reflect also that my own personal faith in Jesus has really transformed me. Jesus changed my life, and he can change yours too. And that's where I would like to finish. Thank you.
Thank you very much, Paul. Now we have about a bit over half an hour for questions, and David at the back has got a roving microphone. I would ask you, if you want to ask a question, raise your hand. I'll point to you, and David will give you the mic, but please wait for the microphone, because otherwise often we don't hear the question. So I have a number of questions. I'm sure you've got lots of questions, including on the things that Paul pointed to, but could I have the first question? Well, we have two to start with. Yeah.
Thanks. I wanted to ask about your opinion on alien life.
Oh, sure.
And how this interacts with your Christian faith.
That's a great question. I've done very little work on what I would call astrobiology proper. I accidentally got roped into an astrobiology project connecting MIT and University of Cardiff about this molecule called phosphine in the atmosphere of Venus. There's recently been another claim about a molecule called dimethyl sulfide in the atmosphere of K218b, this Hycean world, which actually feeds in, interestingly, to some of the models I work on. I'm more interested in how, if we can understand something more about origins of life, it can inform how confident we should be about these signatures, whether they can be produced without life, compared to whether they can be produced with life. So it can give us confidence about these sorts of detections. Someday, if we have a bunch of confident signatures, the distribution of those signatures can tell us something about how life most likely originated. So we can learn both ways. I'm completely agnostic about whether there's life. And the reason is, this chemistry is hard. It's really hard. I try, I'm not really a chemist, but even organic chemists have trouble with aspects of this chemistry. It's really hard to find the brilliance of these reactions goes even beyond the best organic chemists. It's a beautiful thing. So probably origin of life, very unlikely in any single environment with a single experiment. So very small number. Number of planets where this could happen, number of environments on those planets, enormous number. I don't trust myself to take a small number times a big number and get anything out of it besides the number I want to get. And so I can tell you what I hope. I hope there's life everywhere. I think that'd be great. It would help me learn more about how life started, and I think it'd be cool not to be alone. But I just don't know. I think we'll have better constraints on that number in 20 or 30 years. I think more for intelligent life, there's more of a challenge for certain aspects of Christian faith. I don't think these are insurmountable. The Catholic Church, which I'm part of, has thought a lot about these sorts of issues and has done a lot of interesting work in that direction, which if you do want to follow up in the discussion, I'm very happy to talk a little bit about, like, do you baptize aliens, for example? My answer is I find a competent priest to figure out about that. Do aliens even have a religion? I don't know. Do they need a savior? Because we have this idea of Jesus coming to the earth. Did they even have original sin? If they don't have original sin, they probably don't need Jesus, at least in the same way. They would have still a relationship with him, I think, importantly, because he's the second person of the Trinity. He's God, and through him all things were made. But at the same time, I think that a lot of these are open questions, but I think that the aspects of these are, I can't think of anything in particular, and more competent theologians than I mean, very competent theologians have looked into this and have not found any real deal breakers. And so I don't see any real deal breaker. I just see a lot of really interesting questions. Ultimately, questions I'd want to ask these intelligent aliens. When it's just bacteria, which is more the sort of thing I look at, I think there are less questions. It does say something about how likely this chemistry is. It says a lot about how God made the universe, which is why I'm interested in it. It says a lot about how God values life, how precious our planet is in comparison. But I don't think it poses any specific theological challenge, at least none that I can think of. And if you can think of one, I'd love to hear it, because I like challenges.
Second question.
Can I have 2 questions?
Please do.
So my first question is a bit of a follow-up on Aidan's question. So do like the amino acids and nucleotides that life on Earth uses are in any way like special or optimal for their chemistry? And whether like if there exists life on other planets, would we expect them to use like the same amino acids or nucleotides or not? So that's question number 1.
Okay.
And question number 2 is about Luca.
Okay.
Because I know we don't, it's like a concept rather than like a tangible specimen.
Yeah.
But would we treat Luca as sort of a single species? And would we say that in a way it all started with like a single cell or protocell? Or is it like even no use asking that questions given that there may have been like horizontal gene transfers occurring or like competing chemistries?
So I deleted a slide that I now regret deleting. Let me see if I can find it in, I think it's in the Telluride talk. Let me just open this up and I'll show this particular slide. I'm going to answer your second question first and then your first question. So this is metabolism. This is what metabolism of E. coli pretty well mapped out is. Not everything's been answered. There's a couple groups that are doing a lot of great work on it. Complicated. Luca, as far as we can tell, is effectively indistinguishable from this. So it's a bacterium. If we saw Luca, we had it, microbiologists would be able to tell the difference. But in terms of the complexity, there wouldn't be an enormous difference between Luca and E. coli. And another important thing, again, this is outside my own area of expertise. I'm not a biologist. Greg Fournier is a biologist that works specifically on this. He does genetic clock analyses. He tries to figure out what Luca really looked like. He's coming to visit in a few months, so if you're still in Cambridge, get on the mailing list. You can listen to him talk about it. But he's very big on Luca as a population. It's probably a bunch of different organisms that worked together. But it's organisms with a horizontal gene transfer and everything else that at least had enough in common that there's no evidence that anything outside of that population contributed to what it is that we're made of. Doesn't mean that there wasn't other life. Maybe that other life died out. Maybe it just didn't contribute enough that we can find it. We don't know. So there could have been tons of other life, but this is just like there might have been a bottleneck, and we got stuck with a relatively small population of probably a bunch of different species that we call Luca.
If that was a population, do you think it may be one of the, or like a reason or one of the reasons why we have redundancy in like the sort of codons?
Oh, yes. Why the codons are the way that they are is a huge question in Origins. It's actually the central question John Sutherland is currently working on, and a variety of other groups. It could be, and really we haven't gotten much farther than Crick, which is, you could be right, it might be selected for, and it may be that Luca selected for those specific things with the redundancies because there's good reasons for the redundancies. It might just be a pure accident that just got frozen in, or it might be chemically driven, or it might be a combination of those three. We don't know yet. John Sutherland's exploring some of the more chemically driven ways to do it. He's found certain correlations between the codons and particular amino acids. Those correlations are nothing like our own genetic code, so take of that what you will. Maybe he's looking at the wrong proto-ribosome, or maybe it just suggests it isn't really chemical necessity and it could be a combination of selection and chance. There are a lot of good things about having redundancies, though, because when mistakes are made, those mistakes are less likely to really mess you up and get the wrong amino acid in there. So I don't know is kind of the short answer, but a lot of interesting work is being done, especially in that direction. And I think that, more than the actual origin of life, is a more eminently solvable problem. So maybe 20, 30 years, maybe we'll learn something that will blow that out of the water, but it feels like we're getting closer to a candidate solution for that. Going back to your first question, you're gonna, because we've now had a good discussion, you're gonna have to remind me a little bit about what that was.
Yes, sorry. So my first question was about, like, we have, you know, many possible different amino acids and like many possible different like nucleotides, nuclear bases. Are the ones we have in any way optimal?
Yeah.
Or not? Chemically.
They seem to be chemically optimal, but we don't know enough because there's a lot to be explored. Albert Eschenmoser spent a lot of time looking at different ways to do this, something called TNA, which uses a 3-ose sugar, which is a little bit of a different looking sugar. He also did PNA, which actually uses proteins as the backbone. Really cool stuff. None of that seems to copy very well. These use these Watson-Crick-Franklin base pairs, so a sort of hydrogen-hydrogen bond, very weak bond, which doesn't favor any letter over any other, really important for copying information. And it stores information really well. There have been people, Chris Hunter here, a chemist, has looked into different ways, imine bridges doing this. There are ways that he and Jack Shostak have found to do that. They can get up to about 10 letters, so about 10 mers is about the maximum that they can get. That requires a sequence of carefully constructed reactions. It's about 30 reactions, and it kind of falls apart after about 5 generations. And in fact, after one of these research projects was presented, I remember Jack Shostak saying, I think I see why life uses RNA. It just seems to work so well. That doesn't mean that that would be the thing that life always uses, and there are 2 kind of exceptions to that. One is maybe we're just not imaginative about the chemistry, and there's new chemistry we can learn. And there are alternates that, I mean, Jack Shostak's also explored this, where he's changed the genetic code, he's put in different kinds of amino acids, he's used different kinds of bases and sugars. Things seem not to work as well, but they kind of work still, and so you might think in different environments, those could be optimized. If life could happen in a different solvent, say like in sulfuric acid or in methane or something else like that. It's surprising how much chemistry is stable in sulfuric acid, by the way. That's a whole other thing. There's a research project at MIT looking specifically into that. You can't use these because the sugars will just burn up. They just break apart into carbon and water. But PNA, it turns out, is pretty stable. And so maybe PNA would have been the molecule of choice in a different solvent. I think those are the sort of 2 ways in which you might use different chemistries. I think it's very hard to imagine life as we don't know it, and it's even harder to imagine the origin of life as we don't know it, because that's what we don't know about what we don't know. But there are some fun chemical hints, I think, even there. Sure?
There's another question at the back. while the mic is traveling, could I just say that thank you that you're one of the relatively few speakers in the series who actually link their faith to the science.
Oh,
We have had wonderful talks in the past about some branch of science, and then the speaker at the end has said, and by the way, I'm a Catholic, or something like that.
okay.
Yeah. But, and one of our best speakers was an oncologist who actually was an Adventist, and I don't think many of us knew what Adventists were or what they believed, but our respect for Adventism certainly rose hugely
That's awesome.
on that evening.
Yeah.
So anyway, thank you.
Sure.
Could we have the question?
So, has there been any spectroscopy, such as from space telescopes, looking at solar system moons for small molecules of interest?
To a degree. I think probes have been a little bit more effective in this and in a few different places. There was a probe, Cassini, I'm pretty sure, that went through the plumes of Enceladus and got mass spectroscopy of what's in the plumes. There's methane in their plumes. Some people say that's life, so maybe biosignature. I don't really think so for a variety of reasons. Recently, there's been some studies actually showing that there's a lot of hydrogen cyanide in those plumes. And there's some cool things you can actually do with prebiotic chemistry with a different energy source, radiolysis, that mirrors a lot of what this chemistry is. It probably can't get all the way there. For one thing, you don't have a dry surface, and that's kind of important for some of the dehydration reactions, and also concentration. But there's some really interesting chemistry there. There's a whole bunch of complex chemistry we just don't have a good picture for. There may be plumes on Europa. There's some evidence for that. There's the Europa Clipper mission, which will be going and looking to see if there are plumes or cool things on the ice surface that tell us something. Almost guaranteed we'll see something weird we didn't think of. And then it'll give us good reason to dig beneath very carefully so we don't pollute whatever life might be there or non-life, and get a picture for what sort of chemistry might be happening there. For Europa, we know a lot less about what's really happening in the waters. There's a bunch of models, there's a bunch of experiments. There's not a lot of observations. We know something about the surface ice. There's a lot of sulfur there. A lot of that might come from Io. Same thing with Enceladus. There's a lot of complex chemistry. The mass spectrum saw a whole bunch of complicated stuff we can't even identify. That may just be oligomerization of the cyanides and other molecules. So it may not be original to there, but it could be, in which case there's really complicated chemistry happening in Enceladus. I do think those are the best places to look for life, most like the life that we know, because there's water. I mean, besides maybe Mars, because Mars had liquid water early on, probably has some liquid water beneath the surface, had more of an environment a bit like Earth. So, but still, I think that those are wonderful places to look for signs of life and for weird non-life chemistry, which I think is also fascinating.
Could we have the lady in black across there? Yeah.
I too believe that like faith and science are interlinked, but is there a moment in your studies or work where this belief was like strongly evidenced for you, if you don't mind sharing?
Yeah, I'll be honest that my own faith journey kind of worked the other way around. I was a fairly militant atheist when I was first a scientist. I ended up in this research project, the REU research project by the National Science Foundation. Convinced that, I'd read a bunch of Richard Dawkins, and I was convinced there are no good scientists that are Christian. And astoundingly, I think this is a God thing, personally, because this is not statistically representative. Almost every other person in that REU program was a Christian, with a couple Muslims as well, actually. And I got into debates with all of them. I actually had a religious experience at the top of a mountain. I felt the presence of God. That's what really made me into where I am now. That and a bunch of subsequent experiences. I can give my testimony later, but that was really a key linchpin, was standing on top of a mountain, feeling that there was a real creator that put this together, and that creator loves me and cares about me, and that I want to know more about him. I have felt the same thing when I do my research. I don't know enough to know whether I would have felt it the same way if I hadn't had that experience. But there are certain times where things just click so well. Like, if I get out of this and go back to that one slide I didn't have enough time to talk about, and I won't talk about it in great detail. But when I got to this final picture here, where it turned out, when I first ran this chemistry with my friend Oliver Shortle, it was just a huge mess. I made 1 million different molecules. Our code doesn't have 1 million different molecules, it has about 6,000, but I made a lot of that 6,000, like a whole bunch of them. It was a mess. And any code that had richer chemistry than mine made even more molecules. And I also got a really kind of aggressive email from a competitor saying we'd done all of this chemistry wrong because we forgot to account for graphite. If you have a lot of this hydrogen, or if you have a lot of this carbon, these big hydrocarbons, like the big molecule I was looking at, or like one of these, and you heat it up to volcanic temperatures, what happens actually is that all this carbon goes into graphite, or a lot of it does, and only a little bit of the carbon is available to do other chemistry. My competitor was correct, even though it was an annoying moment. I incorporated that. Oliver Shortle and I incorporated it. It cleaned the chemistry entirely. If I ran with volcanic temperatures that are realistic to today's volcanic temperatures, around 12,000 degrees, I get nothing interesting. I just get methane, hydrogen, CO, water, a little bit of nitrogen, maybe a bit of ammonia. But if I run with the temperatures of the early Earth, about 1,500 degrees, all of a sudden I get lots of cyanide, cyanoacetylene, very cleanly without all of these extra things. Really, really nice starting conditions. And when I saw that, I became very prayerful, actually. Another moment where I became prayerful, it turns out this ultraviolet light that we use is much more powerful than the sun. One of the very first projects I did was I scaled it to the brightness of the sun and other stars to figure out where this chemistry could happen in other places. Found out this chemistry can't happen with a hydrogen sulfide, so some of what I showed was a little bit of a lie. We know that can't happen on early Earth. But with a different anion, sulfite, it happens beautifully. And there would be more sulfite there. And it's this competition between the light and the darkness. And all I could think of was John 1, that the light overcomes the darkness and the darkness does not comprehend it. And so, again, I really felt like this was a real symbol of that in this sort of chemistry. Also, it turns out our sun is one of the best stars to do this chemistry, which I think is also kind of a cool thing. It didn't have to be that way. Most of the stars where we find Earth-like planets can't do that kind of photochemistry. And maybe that is part of what makes us special. Maybe not, but it certainly seems special at the moment.
Chris.
I think one thing that we really need to know is whether, if there is life elsewhere in the solar system, whether it had a common origin or not, because it makes an enormous difference to how likely it is for life to originate.
100%.
If we find a second separate origin, then that makes it very likely. And if not, but there is the possibility, if we find out that it all has the same origin, or there isn't any out there, that the origin of life is so improbable that it's happened only once in the universe and we're it.
Yep.
If it is that improbable, I would have thought the chances of actually finding out how it started were a bit low. And on the other hand, the temptation to say God waved his magic wand might arise, which we've of course got to avoid, I think, because of the God of the gaps problem.
Mm-hmm. Actually, I don't have much to say because I agree with most of that, and I think that you've picked on most of the insights. The couple things I would say, and maybe push back just a little bit, is I do think that even if life is improbable, it depends on how it's improbable. So that's one thing, is it depends on the improbability. The other thing is about the sort of God waving his magic wand. Maybe that's true. But then God could wave his magic wand everywhere, in which case life is common. So it doesn't answer the question of how common or rare life is. Because who are we to speculate on the psychology of God? God could wave his magic wand everywhere. And if he waves his magic wand consistently inside and outside the lab, then I can still study it, and I'm fine with it. And if he doesn't consistently, then I am in a bit of trouble discovering this. But that would be true whether we find many instances of life or few. The way in which I think life could be both improbable, and I'll get back to this a little bit. Let's see. There's this picture here. Oh, wait. This sort of thing, there's more things that could have been drawn on this. This is a very simplified diagram. But you see the glycolaldehyde and the glyceraldehyde. If you have those all work in one pot, this chemistry doesn't happen. You actually just get a mess. You actually need to segregate them. There's some ideas about how to recombine these in clever ways. Sulfite actually might be important there, but if we can't, you imagine this as two streams. Let's say you need 3 streams, or 4 streams, or 5 streams, or 6. If you need more than 8 or 9 streams, then you're talking about us being the only life in about 10 parsecs. And that's not many streams. I think we can still learn a lot about this sort of directed chemistry, even if there are many different ways this directed chemistry can be done. If they all come up with these combinatoric problems, these snowflake-like problems, we can still say, look, we've discovered ways in which life could originate, many different ways. They're all exceedingly improbable. So therefore, life is still exceedingly improbable, even though we've learned a bit about it. And unfortunately, what that also means is, because we can't find any other instances, we won't be able to scientifically restrict, like, which is the more probable than the other. We'll just be left with ways it could have happened. They all seem unlikely, and we'll keep looking for more, because chemistry is big. Ideally, life is in many different places, and the ubiquity of it and its distribution would tell us something about how it originated. That's something I hope for, but have no good scientific reason to think is true.
At least you may live long enough to see if there is other life in the solar system. I probably won't.
I'm hopeful. If it's common enough, I stand a good chance. If it's a bit rarer, my children can see it. If it's even rarer than that, we'll just be stuck not knowing, and that's sometimes the way it works.
Thank you. That was a really interesting talk. So my impression is that the way that scientists are trying to kind of study life is through this very materialistic perspective. I'm a scientist myself. I'm an embryologist.
Oh, cool.
I personally believe there's something beyond the physical.
Mm-hmm.
So I'm just curious, are there any scientists looking at, I don't know, some kind of like energetic form of life? Because just because you have a vessel, and that, there's the perspective that, from a scientific point of view, that we're just bags of chemicals and we're just something physical. But if, just because you have a vessel, like, a phospholipid sphere, does that mean that you have life? I disagree with this.
Mm-hmm.
And so, yeah, I'm just curious, like, are there people looking at the emergence of some kind of soul? Do people study how this emerges? And I know that there are biologists. This is a very early field. It's bioelectricity. So there's a guy called Michael Levin. He's based in...
Oh, yes.
And he's studying how electrical fields... you have DNA. They show that gives rise to the hardware, i.e., the body, the physical body of an organism. But the energetic, the way that our morphology is created, so like where an eye is placed, that can be programmed by electrical gradients, and like how you have voltages distributed throughout the body. Incredible. And this is interesting because it gets to more like quantum physics and electricity. So I'm curious, like, I really do think in the next couple of decades this is gonna like really blow up, because it changes the way that we see life, going from just like a physical chemical bag of something to something more energetic. And I'm just curious, if there are people in your field studying this kind of the more energetic side of the chemistry and how that emerges.
Yeah. I think that you might have to be a bit more of a philosopher to say, like, how does information fit in with a materialistic frame? So even without all this electricity, I think that's incredible. I want to learn more about it. I don't know enough about it to even comment, except that I just want to read more about it. And maybe we can discuss it. You can suggest some things I can read. But even setting that aside, even if it is just these molecules all linking together in this way where you have the ribosome, which is just this enormous machine, which is connecting amino acids to to nucleotide trimers. That can't be adequately explained just in terms of even quantum fields. Even if I introduce an electric field, that's not going to do it. There's an informational component. There's a code. There's something about that which transcends. As a physicist, I think that's true for water, actually. I think there's ways water behaves. Water's an incredible solvent. It has 90 outstanding mysteries people haven't solved, people don't understand. I met a chemist a couple weeks ago in this Origins of Life as We Don't Know It meeting. All he studies is water. It's incredible. There's a form to water. There's something mathematical. There's something mathematical about how planets move around the stars, which cannot be about matter. This actually gets to one of the things I was asked near the beginning, is maybe I can elaborate on what I mean by Platonism. And this gets at that idea, which is that I think that there are these forms. I don't think they live in a separate realm because I don't think they're physical in that way. I don't know how to make sense even of basic physics. If you can't answer, what Stephen Hawking even said about it, what breathes fire into the equations. There's something about that that's not physical. The number 2 is not physical. But we have better empirical evidence that we should believe the number 2 than that we should believe electrons, because we use the number 2 a lot more in physics than we use the idea of electrons. So I definitely think that there is a non-material component, even for good scientific explanations. I think there are things that go even beyond science, the first-person perspective. Science is always third-personal. There's a great philosopher, Lynn Ritter Baker, who did a bunch of great work on this. You can't break all first-personal statements into third-personal statements. This individual and their decisions can't be broken down into basic scientific statements. So there are aspects of that science just cannot tackle. And that's critical for all science, because science is run by humans. I make a decision about which experiment I decide to perform. And so I can't then incorporate everything into a single field. There's one last thing that I'm going to say, which is a sort of tension that I see among a lot of atheists, materialists, who will say, you know, everything's just matter in motion, all random. And they will also still entertain, in the singularity, if you know what the singularity is, the great AI revolution, will all get downloaded into a computer. Well, that's horrible. I'll be dead, because I'm just this matter. If I get moved to somewhere else, what is me? Me can't be matter anymore. Same thing with the idea that we could be in a simulation. Lots of people entertain that very seriously. If we're really in a simulation, then it's not even just materialism is false. Even the dualism picture is false. We're really in an idealistic world where it's all ideas and there is no matter ultimately. And so I really think there's so much that can't be made sense of, and there's so much that seems intuitive that just does not come down to materialism, even for purported materialists.
Shall we? I think we'd better say 2 more questions and then we finish. There's a gentleman over here.
Thank you. That was an excellent talk. Unfortunately, I'm rather weak in chemistry. I'm just curious, is there some way to put the chemical discussion that we've been having about life, the chemistry of life, into the context of, say, a book like Just 6 Numbers by Martin Rees? You know, maybe we have more than 6. Maybe it sounds almost like we have 50 numbers here, but it just seems like that would be beneficial to someone like me.
I think that's great. And there may be even better suggestions than the 2 I'm going to give. One is right there, Is Earth Exceptional by Jack Shostak and Mario Livio. Its ambition is to do what you say.
Good.
I would say Jack puts a little too much chemistry into it. It will be a bit of a hard slog in the middle. He just got too excited. I think that's what happened, is he was working through it. It's such beautiful chemistry. He started drawing all these diagrams. Right. But it's still quite understandable. And if you skip to the second part by Mario Livio, he's an astronomer. He talks a bit about this from an astronomer's perspective. It's easy to understand, simple, clean. The other book that I would recommend would be, even though I spoke badly of Richard Dawkins, I do think he wrote an excellent book on this, which is called The Greatest Show on Earth, which talks about how evolution works. It's a bit of a myopic perspective on evolution, but he's very good at talking about all these components in a way that anybody can understand. He's a wonderful science communicator, really.
I just want to say one other thing. This young lady talked about what profound experiences, and I had one about almost 2 years ago when I saw, maybe other people haven't felt this way, but I saw the total eclipse in April, and that April, I think, was 2024, and it was profound because it occurred to me that as far as I know, no other planet in the solar system has a total eclipse or can have a total,
Yeah,
a
you are
moon
absolutely right about that. There's no
covering-
other planet that can do it, and we
it's
already know
interesting.
that for sure.
As far as we know, we're
Yeah.
the only life that can appreciate
Yep.
that.
There could be
It's
planets in
almost
other solar systems
Yeah.
that do this, by the way, but in our solar system, given the planets we know,
Yep.
all of them just have moons that are way too small or themselves are way too big. It's astounding that we have the moon that's just the right
I mean,
distance.
it's incredible.
That blocks
It's
it just
incredible.
perfect, and you see the edge, and it just excites the imagination. Absolutely,
Yeah,
100%.
it was absolutely remarkable. Like God is telling us, hey,
Yeah.
just a little something for you. It's not because it's not necessary.
No.
Yeah.
No,
It's like signing a painting.
Yeah, there's no good reason I can think of, even from a sort of fine-tuning of life perspective, for that to be true, and yet it is, and I love it. And yeah, I saw one when I was a kid. I was supposed to see that one. I was on the path. It was cloudy. It's a bit sad. I'm definitely going to try to see one again. Once you see a total eclipse, you're going to start hunting for them. It's just what happens. I love that. I love that.
One last question.
Very quick one. What about the laws of physics? Sorry.
What about them? What about the laws of physics?
Well, you mentioned once the laws of physics in your talk, didn't you?
Oh,
I mean,
yes.
where did they come from?
Yeah.
And are they the same everywhere? Could they be different? So that was the only thing that was going through my mind.
Yes. So Baruch Spinoza says the laws of nature are always and everywhere the same. I think that's almost a sort of rationalist statement of faith. And there's a sort of physicist statement of faith that works with this, but it's really a statement of faith. It can't be empirically proven. And that is that the laws of physics are the same today and tomorrow. They're the same here and on the moon and elsewhere. And they're also the same no matter which direction I'm facing. Emmy Noether, brilliant mathematician, showed that every single one of those symmetries, every single one of those articles of faith involves a conservation principle. So if the laws of physics are the same today and tomorrow, that means energy is conserved. If they're the same here and on the moon, that means momentum is conserved. And if they're the same no matter which direction I'm facing, that means angular momentum is conserved. You cannot prove any of these conservation principles because every experiment I run shows that energy isn't conserved. I'm always losing some. Now, I believe that that's going into random motions of molecules, and that still makes sense within the grander paradigm. But proving that would be almost impossible. And even if somebody could run this amazing high-energy physics experiment showing that energy was genuinely lost, their first thought wouldn't be, oh, well, so much for the law of conservation of energy. They would think, oh, there's some new particle or subspace or something else where the energy has gone. There's no way to definitively test, at least as far as I think, those sorts of things. Where those laws come from? I have a belief that might be heretical, in which case if the church tells me that, I have to change my mind, I suppose. But I think those laws of physics are eternal. I think that they're part of those forms, just like the number 2 and circles and everything else. And it's God who takes among all the many laws there could be, those laws, and connects them to our own universe.
Thank you, Paul. Paul, I have to admit, a lot of the science is above my head.
I'm sorry.
And I'm possibly not the only one here. But as a linguist, I'm a passionate linguist, and what I love is the complexity of language, words, syntax, and to see the multiple examples of different languages around the world is just fascinating to me. And I see you have the same passion for your subject, and long may it live.
Thank you.
And I think what we share is that enthusiasm, that awe, that respect for the amazing complexity of God's creation. So we have something in common, even if I don't understand.
Amen. Well, I probably don't understand what you do either, but I'll work to be more understandable in the
I
future.
could talk for 3 hours about that.
Excellent.
Anyway, thank you very much. Let's.
Charity that works with autistic people, normally youth, and tries to provide money for autistic people, especially people who are far along in the spectrum, to have things to do, to develop their social connections, to learn to be compassionate in things like sports or other kinds of activities. And so it works with the council. It provides certain money and other resources for families so that they can have their children do things with other children and learn more how to integrate fully into society. I'm very passionate about this because one of my children is profoundly autistic, and it's opened my eyes to both the challenge and the beauty connected with that particular condition.
So do give generously.
Thank you.